I'm going to talk today about protein interaction, or the prediction of the pairs of protein interactions. Next week we will get into, or actually from Thursday, there will be an overflow of this to Thursday, and then we will begin to talk about the prediction of the effect of single residue invariance. Then will come the lecture from Jana Schmidt's group, not 10, that's 19th. A mistake here. That's next Thursday, and that will, in fact, the 19th will also be the last lecture, and then we will have wrap-up. Wrap-up means essentially you come ideally with questions, and we will discuss questions for the exam. Okay. Overview over the so protein interactions. Typically, the way you see protein interactions these days are as these large hairballs, where what you see is the connection of each protein with all the others. The background idea really is the idea that the cell, and that is illustrated in some sense here, that the cell is much more like a clock with cogs filling the space, and every cog in there interacting with another cog, then like a bag of water. It is full of interacting pairs. Every protein somehow interacts. What you see here is cascades of interactions. What you see is pathways that all interact. That's tryptophan metabolism, so to make tryptophan, digest tryptophan. And all these networks begin with pairs of interacting proteins. Again, when I'm talking about interactions, please don't feed her. I understand you didn't. It's just, thanks. Yep. Not association. So protein interaction for us means physical touching of two proteins, not A activates B, activates C, and then D interacts with A. This is not an interaction for me. Prediction typically is considered the acid test for understanding. What about machine learning? So if I sort of have a statistical formula that tells me that this means something, unless I can predict with it, it is really not true. But in machine learning, we sometimes can predict, do we have understanding? And the regular answer to that question from many is that in fact some machine learning is just like black magic. It's a black box. It may extract rules, but these rules are hidden in some box. Is that true or not? So again, let me sort of go through some ideas of machine learning. So you have a data set, you split into a training set. For that set, you sort of optimize some parameters. And then you have a test set. The test set you hide under the table. You don't use for optimizing anything, for developing the methods. You cannot choose the way you split these two sets at random. You have to remember that there are biological realities that you have to choose between testing and training. You should not be able to do homology inference, for instance. So the similarity between these two sets have some constraints. When dividing the sets, ultimately you have to make sure that every protein in your data set has been used for testing once, so you have to repeat this, in this particular case here, 4 times, so in the way I show it here. And this is not enough. We need a threefold split, where we have a training set, a cross-training set, and a testing set. Why do we need to do that?
To prevent overfitting?
That is true, but essentially the idea was, again, for most publications of machine learning, such as the weaker package, they get away with training and testing, so they don't have the step in between. And they do cross-validation, this is the way you see it in Wikipedia and everywhere else, to prevent overfitting, to prevent overtraining. So for the two-way cross-validation, your argument is used. So why then do I need a third? Although you're right. What is it that people typically overfit still? What is it that test sets often are used for that they shouldn't be used for? Do you remember? So one thing that they're used for very often is to answer the question, is linear regression better than SVM? So what type of machine learning? Or in the machine learning, the SVM sort of trains the split of the data. But then the question is, should I use that as input or that as input? And that question is typically answered by looking at how well does it perform for the test set. That means the test set is used to make a decision. Whatever decision you make, that's part of training. Even the decision, linear regression versus SVM, right? And that is exactly what you need the cross-training set for. The level of, let's call it decision making. Setting some free parameters. And that is really an important thing. And you have to get that right. And you have to get in this still the overlap right in the sense that what is in here and is in here cannot overlap, for instance, on the level of homology inference. The same is true here. Okay? Not that easy. Now, in order to make that clear, you somehow have to group proteins into families and make sure that there's really no overlap. Now, that still is not enough, arguably, because after all of these, if you did your cross-validation right, you still may have benefited from some information that you had while you prepared your method. Yes, you may have been very, very clear on never looking at this, but you knew the answer for that. Your algorithm knew it, and maybe some of your decisions, although you did not really you were not aware of it, were driven by knowing these data set. So what we can do in order to avoid that this is a problem is, how can we address that problem? You have developed a method, you have done machine learning over the course of your thesis. The thesis is 6 months, takes 1 month to write it up or something like that. So you have, after 4 months, you have the results. Now what's the last thing you can do in order to prevent that? Last problem. But there's some gazing, some over-optimizing from the knowing the test set. What could you do? Anything else you can put in? Any idea? So you've worked on your topic for 4 months, whatever machine learning topic it was. Well, you can, do you know the answer?
The thesis and then go back and take the data that has been discovered in the time that I've written the thesis.
And you're saying that because you remember that?
No, we're doing the Kafka challenge, does it?
This is my dream answer. I'm yet waiting for the person in my group who is really going to do that. But yes, this is exactly what you have to do. In fact, we have done it in our group at various points. Just recently, in a paper for text mining, Juanmi has done it, and the paper was written up, and then we essentially went back and did another test in that particular case. Not only did we sort of redo something on a new data set, but we did a completely different test. And that, in that particular case, turned the manuscript, and in fact, in some sense, turned the field. Or may be turning the field. Yes, you ideally go back and have some set that is like a pre-release or latest release or the last 5 months results that came in, the last things in the database. You have to make sure that these, again, are independent of whatever you have used before. So you have to use homology inference distant ones, or things that you cannot use for homology inference from the latest new set. And then see, that will be a small data set. Most likely. So you will not get statistically significance, but you will get trends. And it is important to see whether essentially what you estimated on the test set is the right ballpark. That you can see here. You can see whether you have been grossly wrong. Or ideally, actually, you're doing a little bit better for that set. And then maybe you can write that in the paper and come up with some ideas why maybe there's something easier in this set, in the ones that, but it clearly gives you good material to believe that you were right. Now, if you did this and the latest pre-release set, you're doing at least as good or even better, so you're essentially getting exactly what you estimate, then you can really trust that the machine learning device extracted some principles, right? Here's another story I talked about before Christmas, this story about the hotspots extraction. In the hotspot extraction, so what we're predicting here from sequence is which residue is involved in protein-protein interaction. Purple is predicted, red is observed. And I said that the system essentially learned to predict hotspots, although it had never been trained. That's another example where you, after the machine learning task is completed, this has nothing to do with accuracy, performance estimates, but what you learn is you extract the biophysical principle you never put explicitly in. So the system clearly learned a rule, learned some feature, in this particular case the feature of that the hotspot exists, or that some residues are really important for protein-protein interactions. Now, this is still not enough, because now you have to do some statistics on data sets. So we talked about that before. You have to see the results that you have with respect to random. That's one thing. So this method may be best, but it actually, first of all, it's obvious that this method really is close to random, but also still missing is the idea about the standard error. What do you need in order to work out a standard error? Well, you need the distribution of performance for a data set. Say the method that you developed were to predict subset localization. How can you have a distribution of subset localization predictions? So you have a method that predicts, let's just look at the simplest problem, or we can even, subset localization is a good one. So we are looking at the simplest problem. You want to predict whether a protein is nuclear or not. How do you get a distribution from your prediction method? And the obvious problem is you either get it right or you get it wrong. So distribution, all you will get is some set of proteins for which you are wrong and some set of proteins for which you are 100% right. And your accuracy is determined by the sum, by this over the sum, right? So just for the sake of the argument, if that were 35, oh no, I'm getting into numbers that I cannot easily compute. If that were 20 and that were 80, then your prediction accuracy is 80%. But how does that give you a distribution? That's exactly the number essentially I write here. How do you turn that into a distribution? Standard error. Again, in order to compile a standard error, you need to have some standard distribution, or some function like that, where what you see here is number, and what you see here is the Q2 that I'm plotting here. And typically, what the number is that we see here in this particular case would be 37, would be the average. The standard error has to do with the standard deviation, the sigma here, and the number of samples. So the rule of thumb, the standard error is the sigma divided by the square root of n. So this is the proportional signal, roughly, right? But where do I get the distribution from? I have n, but I don't have sigma. Is it, again, trivial, or? Was she the only one who's telling me whether it's simple or not? Is it complicated or not? It's something that we talked about. It's a repetition of, it's a lookup in your brain, or wherever it is written, or a book. So no, you don't really know. You do know?
Yes.
Yes.
We get some bootstrapping.
So it's too trivial. Can you do me the favor?
Sometimes I know it, but yes, I don't know.
But that's the right thing. There are different ways of doing science. Clearly there are the scientists who just sit down and think and work it out, and they do right. And some are more like I. So I'm not intelligent enough, so I have to try, and I have to persistently try, and I hope that I will be intelligent at least enough to see my mistake. In order to do that, you have to always just jump. First, begin by jump, and then look at it and see whether you see a mistake in that. I feel this is often helpful. Yes, so The way you do bootstrapping essentially is you put all of these into your data set. In this particular case, the data set, you have 100 points. You take out 50 and compile for the 50. What is your performance there? You take out another 50, what's the performance? You take out another 50, you do that many thousand times, and then you get a distribution. This is not the real standard error, but it's an approximation. And again, depending on how you do this bootstrapping, so again in Wikipedia what you will find is take the 100 and randomly pick out and put back. This typically overestimates or underestimates your error. So taking a smaller set and not putting back typically gives ultimately a better estimate for in practice a better estimate. At least for the field of computational biology. Now, then we have a standard error, then we have a standard deviation, and that is important in order to see whether these methods here differ. And maybe what I'm doing today is trivial, but again, this will be part. It's not unlikely to be part of an examination. How good is random? How good are the best state-of-the-art methods? Are they tested on the same data set? Again, implicitly, when we see tables like that, numbers put next to each other, you immediately believe that what the authors mean is they were done on the same data sets. Most of the time, this is actually not the case, because the authors look that up from publications. And in publications, people use different data sets. So most of the time, in fact, those tables, certainly the ones that use other methods put other methods into the fold, do not use the same data set. And that is sort of apples and oranges and should never happen. So you have to really test on the same data set, see where random is, see where the top is, look at error estimates. And then in this particular case, for these error estimates that I have, the question becomes, is method 3 the best and performs at 37% accuracy? Is that a correct statement? So the answer in this particular case, I would argue, is no, it's not a correct statement, because even by one standard error, I cannot distinguish between method 2 and 3. So method 2 is one standard error. It's actually is a reliability or a statistical value. One standard error, it covers 90% of the distribution. So at 0.1, at a p-value of 0.1, even at such a small p-value, this could be 36. And at the same small p-value, this could be 36. So plus minus 1 gets you to, the minus 1 gets you to 36 here, the plus 1 gets you to 36 here. Within one standard error, they're not distinguishable. So therefore, I would call this statement not quite correct. It is true that numerically is the highest value, but it's not distinguishable by even one standard error. And whenever that method would say method 3 is best, it shouldn't say use the word best in my view, but simply should say numerically is the highest value, although we cannot distinguish, or something like that, an addition like that. So now comes the question, if we can predict, and if we have separated training testing set well enough, does that mean that machine learning now has understanding? My argument is, in fact, machine learning is not black magic. This is a complete misunderstanding. And the complete misunderstanding typically comes from the fact that too often is done in the wrong way. Now, the real issue, however, is when people extract the rules, is that the rules become so complicated that we cannot understand them. And then it's very easy to say, okay, because these are complicated rules, they're not helpful rules, hence the machine has extracted some knowledge that is again black, as a black box, because I cannot understand it. And my argument here is, no, that's not the case. What is the case is that the problem that we have is so complex that we cannot formulate it in simple rules. This is not E equals MC squared or something like that. It's not the type, it's more a chaotic problem. It's more the type of problem where we don't have simple formula. And when we don't have a simple formula, the best we can actually do is to have a machine learning device that captures the complexity of the problem. No black box. A very open box. And you can learn a lot from this box. And I consider this black box story for a mistake, again, that comes because most of the time people have problems to do training testing right. And since I say that most of the time people have problems, I'm going to show you a few examples for how complicated the separation between training and testing becomes for the problem of predicting protein-protein interactions. Again, we want to predict that protein 120, BGP 120, binds the antibody CD4 here. The original idea was really to look at consecutive residues on one, consecutive residues on the other, and train these segments that are in contact in some machine learning device. Say here they contact, outside they don't. And simply try the machine learning, to develop a machine learning device that distinguishes the grayed out region here from all the others. And that didn't work. We developed all kinds of false positives methods, such as the prediction of subcellular localization. And then we got finally to this issue of putting all of these together and going to the pairing problem. And then we got into the following problems. Here's the first problem. The first problem has to do with redundancy reduction. So I said that when we do cross-validation, we have a training set, we have a cross-training set, we have a testing set. Very, very important, but let's simplify again. Assume we have only training and testing. And in order to separate them, we know there must be a grouping into families. Because ultimately, all of these here are somehow similar within homology inference reach, and they cannot, any pair in here can only be part of one of the sets. They can only be part of training. But you cannot take one out for training and one for testing because they are trivially too similar. Okay? So that's why it's some grouping like that. In many cases, we would pick one representative from each family. That's the way you prepare your K-for data in some cases. So there's a simple family clustering, and this is relatively trivial. But for protein interaction, what we actually have is not A, not protein A. What we have is AB. So what do I do if this here is AB and this is AC? Where in this particular case, the way I sort of mean the colors here, B and C are different colors. Meaning that A interacts one with B, one time A interacts with B, and one time A interacts with C. A pairs with B, A pairs with C, that's experimentally known. But B and C, the H-val or the PID is less than 0, for instance. So B and C are so different that just looking at their sequences, I cannot say, well, if A binds to B, I assume A will also bind to C. Right? Then B and C are different. Okay? It's the same A though. So can I put the same A in different clusters if the A interacts with different proteins, different binding partners? And originally that was done by Yonki Park and Edward Marcotte. And essentially what they did is they looked at 3 different cases. The first case is that when I predict the interaction between A and B, both A and B had been used for training, but not the interaction. So A had been, for training what we had seen is A pairs with C and B pairs with D and all kinds of things, but not AB. So that's case 1. I want to predict AB, and both have been used in training. Case number 2, one of the 2 has been used in training. Case 3, none has been used in training. Not the interaction, mind you. Okay? Should it matter? Should it? Do you expect a difference? Let's look at the most extreme cases. So 1 and 3. And I'm not even sure that I show any data for in between. If cross-validation is right, correctly done, in principle we do not expect this to differ, because A interacting with C is just something else. The A interacting with B. And what they clearly saw is decreasing performance. So it's much easier to predict an interaction between 2 proteins if both of them have been part of the training set. Okay, in terms of numbers, so here for two different for two different methods that use only sequence information to predict protein-protein interaction, pipe 2 and SIGPROT. You see that for both of those three, where both of them have not been part of training, class 3 is much lower, green is much lower than class 1, where both have been in training. And the class 2 is somewhere in between. That came as a surprise. But this means that in fact now I do not only have to classify according to the single proteins, but even the pairings. Okay? So we have a new kind of redundancy reduction. Ultimately, the way you do it in the method is the old way. You reduce on the number of proteins. But this is surprising given that you have actually pairs here. But now the next question becomes, do you only have to do that on the positives, A binds B, or also on the negatives? So the negatives are essentially, how do you produce the negatives? So if we start with human, about 20,000 proteins, and then I have a bunch of pairs of all human proteins and their pairings. And for all of that, say I have 50K. I have about 50,000 interactions between all the human proteins, so possible are 20,000 times 10,000. And of all of those, I have good experimental evidence. I have experimental evidence, I believe, at some point. Now we have experimental evidence for 300,000. I have good evidence for about 50,000. I do not have many experiments where I see that A does not bind B. How do you create the negatives for machine learning?
Choose random pairs which are not in the positive set.
So we look at a subset of all the ones that have not been observed. And for the set that has not been observed, although I say positives are 50K, I certainly take what anybody has ever seen. I don't care about the accuracy of the experiment. As long as anybody has seen it, I exclude that. So here in between there are 250K that fell out. I would just ignore them. They will not become positives and they will not become negatives, right? But as long as nobody has ever observed it, here, so I will apply this rule, Simon's rule for these, right? The ones that have never been observed by anybody, and then possibly a random subset. Okay, a random subset. Now, this brings us to the next issue. So the positives here are 50K. The negatives are 200 million minus 300,000, so essentially 200 million, right? A subset of 200 million. So obviously I cannot do 50,000 against 200 million. So I need a random subset. But how big is that subset? What number should I choose? Anybody has any idea? So this is not a simple, this is not something where so we don't have a solution, and any solution you can come up with is as good as the solution that any expert has. There is no good idea. So ultimately the idea that people have is we somehow sample according to what we expect. So what is an average number of interactions that a protein has, or something like that? And that is what we use as a base. So one number that comes often in the literature is 100-fold. So for every positive, we use 100 negatives. But they use 10, 100. This is all 80. Or the field has not come up with a good idea. have a large set of negatives, and we need to keep the imbalance. So in nature, we do not actually know how many proteins a protein interacts with. We do not know whether each human protein on average interacts with 10 or 100. We believe that the real answer is somewhere in between these two. But again, there are arguments that the number may even be larger. So it is unclear. But we do know that the number of negatives is, or the non-interacting is clearly larger. So we believe that in nature there is an imbalance, and we want to keep some imbalance, and we put the number 100. Or whatever else you want, right? This is a hack. Now, back to that question here. When you go to the literature, this is the first way you try to answer is you try to Google. You, in fact, try to see what people use when they develop methods, and they all did not consider the negatives. And the reason ultimately is because the way it's sampled, you're not using experimental data for the negatives. You just say everything that has not been observed. And in some sense, it's a simulated set. It's an in silico set. It's a set produced by the computers. There's no experimental evidence that says that this large number of proteins is not interacting, right? So, because it's sort of an arbitrary set in some sense, people did never consider really to do a redundancy reduction on it. And to really, in terms of cross-validation splits, consider negatives, because negatives are in some sense not real, but they are needed. And this is a reasonable way of doing it. Okay? What this plot shows, and again, I'm sorry for the complication of this plot. The take-home message, however, is simple, and the work is done by Tobias. It's a recall precision curve, and this precision essentially is a delta. The delta is between doing a filtering on the negatives or taking all negatives. And you see that for most methods, in fact, whether the negatives are filtered or not makes a big difference. Okay? It turns out there's a reason to explain that, but the important point here really is all of these lines here are not zero. Whether you filter or redundancy reduce on the negatives or you don't, matters. It's not really clear that it should. It may make sense. What I don't show in this curve is which of the two is right. That is a longer story. Ultimately, what comes out of that story is we have to really also consider the negative interaction pairs in order to cross-validate or choose the cross-validation sets. Now, this implies, in order for redundancy reduction, let's not go through this in detail, but it gets much more complicated. Now comes the next issue, and that has to do with data. And let me cut this one here and just make the simple point. The 50K after redundancy reduction, they melt down to numbers in the few K category. So few K is absolutely clear is too small. We don't have more. This is all we can use. But since we have so little data, should we take all the data? The first question is really, I say it's somehow clear that this is not enough data. How can you figure that out from the machine learning device? Well, one reason is you could argue my prediction accuracy is not very high. And that is because I don't have enough data. But that could also be because you put in the wrong input data, or you made a mistake, or this could have all kinds of reasons. But how can you distinguish between a situation where you simply do not have enough training data and a situation where you may have enough? Again, is something that I talked about. So in some sense could classify as trivial. It's not, but it's very intuitive, yeah? I'm sorry that I did not imply. Oh, I misunderstood your gesture. I thought you meant it's very simple, that it came out of your head. It's very simple. You can. This is the kind of wheel that is easy to reinvent. Just trust your intuition. How could you do that? Well, one way is simply to half or double the data. So you have a data set, say in our case it would have been 10K, you just take 5K. And the first question that you have to analyze, is there a difference between 5 and 10? If not, then it's clear that you have already, whatever problem you have, whatever level of accuracy you have, but the number of data points may not matter because from 5 to 10 you didn't jump. You didn't benefit, right? If there is a big increase, it does not tell you that you don't have enough, but gives you a tendency. So then you can sort of approach that. Is there still a jump between, in your data set, between 9 and 10? If that answer is yes, then it's very reasonable to expect that between 10 and 11 there will also be some increase at least. If you add in order of 1,000 points from 5, do you sort of reach a plateau? Or are you still in a distribution part where you see this clearly increasing? And that at least gives you a clear direction. Yeah?
Could not also be the reason that maybe you're pretty close to random, and then there's no difference between 5 and 10, and the reason is both are just not enough, and
that is a very good point. So you have to make sure, that's a very good point. So you have to make sure that your prediction is not random.
Yeah.
That's absolutely true. Yes, good point.
How many percentage points of accuracy is not random anymore? Especially with a smaller data set.
So let's turn that around. Let's assume I were a factor of 2 away from random at 5 and got a factor of 4 away from random at 10. Is there anything else that could go wrong? Could there be any other explanation other than is really my increase in the data set size helped because I got more examples and I needed more examples? Is there anything else? I couldn't think about anything. So this would not be... So yes, you may still have a problem in your input space, and that may be the reason why your accuracy is lower than something. But you clearly know for your solution, that is for your input, the data set at that point is not big enough. Again, if you had a different input, that may change. So you have to test this on whatever you believe at your data set is the best input. And typically one example for that is whether you use alignment information or not. You typically need much fewer data points when you have alignments as input. And that again is shown as a delta here and a little bit more complicated. Now, here we have another issue. And again, instead of showing the original data, I should possibly go to the overview slides. What I show, again, now I have a delta in the precision, and what I analyze here is if I have A interacts with C here and A interacts with D and C and D are totally different, okay, I have to be careful for the cross-training. But at least within the training set, should I use both? Assume both are good data points, good experimental data points, and suppose that C and D really are different proteins. Should I use both? And the difference is negative if using both is a bad idea. And you see that for several of these curves that you see, for several of the methods that I show, the difference is negative. Meaning that in fact, yes, you may not have enough data, but nevertheless, you should only use one per protein. Which you may call is sort of contradictory or surprising, actually is, but it's a finding. And in some sense, all of that adds up to the conclusion that cross-validation for protein-protein interaction prediction is really complicated. Now the third challenge here is how to do it best. And how to do it best, there are essentially two different methods or two different ideas. One idea is you sort of take your protein and you test it in different conformations and you essentially move it around. So you essentially, like the sliding window, you try to find, in a docking sense, try to find a way to make it fit best. Now, you put in a feature, the approach 2 is you put these two proteins into a grinder and you somehow look at average features. Could you think about a way to do the second one here? I'm not entirely sure why I call that step 1. Say we would go into the binary classification and just say A binds B. Not where it binds, but A binds B, it does or it doesn't. We would think about the grinder. What kind of methods could we use? So all you need to use is somehow the sequence information of protein A, the sequence information or the alignment information of protein B. What kind of methods could you think about that could then possibly predict protein pairings? That has been, think about any, throw anything at me. It's something that we have discussed before in different contexts for subcellular localization. So remember, for the protein interaction site prediction, I have running windows because I want to see which residue binds, right? Now, essentially, you want to predict whether A binds B, not where it binds. And the reason why we want to do that is because when we try to predict where it binds, I call that method 1 here, the sampling here around, somehow in our hands that didn't work. We got somewhere when we knew that they bind. So that's why we sort of addressed the binary prediction problem A binds B first. And that brings us back to the question. Any idea about a method that could do that?
Use amino acid composition or use surface composition?
Yes. And now what you essentially would look at is 20, you would describe your protein by 20 units, both proteins by 20 units, and then you would hope that you capture in the correlation between amino acid composition between two proteins, whether they interact or not. And that is the simplest thing you can try. Do you believe it's going to work? No, because of the way I ask or because of other reasons? So it's not going to work. Sorry, we tried it. It didn't work. The definition of didn't work is in the ballpark of random. Why could that be?
If you look at the surface composition, you take into account the whole surface, and if you have a binding site, normally you would expect that it only binds a fraction of the surface.
That's exactly, so visually it's exactly this problem here. So most of the things on the surface, they don't bind. So most of the surface correlation, in fact, in subcellular localization, the entire surface sees the environment. Here, most of the surface would not correlate in between the two, because most of the surface would actually not bind to each other. It is only whatever fraction of the surface here, whatever fraction of the surface here, but it's a small subset. And that, in fact, is exactly the problem here. So how could we do it?
I would go for motifs.
Yes, motifs is one idea, and the motifs, and the way we did it here is the profile kernel again. So the profile kernel that was also used for subcellular localization prediction, where you essentially look in this example for the profile kernel with size being 3, so 3 mers. We look at 3 mers in the alignment of protein 1, and we simply count all the possible occurrences of the particular k-mer throughout that alignment in that family. We did that for the second protein, and then we simply crossed these two vectors. Not that simple, but essentially this is the kind of thing that takes a while to work out in detail, but it's doable. I mean, to get to that point took more than a master's thesis. If you knew that this is all you need to do, then this is clearly doable in a master thesis. Yes, thank you. Okay. Now, this brings us back to something I believe I may have said in passing. When we developed the subcellular localization prediction method, we were guided somehow in these K-mer and these motifs, in this thinking by the nuclear localization motifs, where it is true that one particular region, if that put onto a protein, determines that this is going to be a nuclear protein. And that got us to the question, can we essentially extract the K-mers that are these nuclear localization signals? And that brought us to the question, how many, if I make a classification for subcellular localization or protein-protein interaction, how many K-mers are actually used? Any idea? So essentially we're looking at most informative motives. So we assumed that it would be a few. Maybe we would get the nuclear localization signal, the one. That's what we had hoped. And we found, in a set of 10,000, we found 10. Where this actually worked. But 9,990, it didn't. And for most of those, in fact, the numbers were, this is sort of the box here, this is 200,000. K-mers for making one, classify one protein, okay? This is very far away from one. And so what this one shows here is the percentage of the total decision and the number of motifs, or percentage of K-mers. So here, 50% essentially is covered roughly by almost 1,000. Only 50% of the decision is made by the strongest 1,000 K-mers. So not only is the number high, but the Gaussian tail, if you want to call it that way, is really contributing a lot in terms of information content of the, so you cannot zoom into the strongest one. You can, but you actually only capture 50% of the information. And the same is true here for the protein interaction. So it's really a complex set of K-mers that is used. But ultimately, the method works better than anything that exists. The new method is in blue here. This is for proteins that, in fact, now since we do have to distinguish between proteins that have been used for training and proteins that have not been used for training, so there are two different curves, two different sets of methods underneath. And again, so it's thousands of motifs. Let's talk about other ways of predicting it. So one simple idea is the duplication event. So you have an organism P and an organism C, and in the duplication you essentially duplicate the interaction. Meaning that if you just, in both cases of organisms, you looked at the entire set of proteins that exists and say yes or no. Now, what I mean here is protein E, but essentially I mean family E, right? Anything that is sort of similar to protein E. So the X means I have a member of that family. Okay? And from this vector, from the similarity of this vector, you can essentially infer duplication events, and that allows you to predict protein interactions by crossing these vectors. And that is a method that is out there since a while, and is one of those remarkable moments in history where two groups published it in the same year, completely independently from each other. So I know all the details about both stories. They're totally independent. They reinvented the same idea at the same time. It was somehow in the air. The idea two is the domain copy idea. So that's simply here. You see that in one organism you have these two domains. In another organism you have these two domains as two separate proteins. And one suspicion why you see something such as this is because these two interact. And that's essentially method two. And again, was developed independently by two different groups roughly at the same time. The next one is correlated mutation. Some of you may have heard that before. Comes to us from Thomas Hopf. Originally, this slide also comes from Thomas and Debbie Marks. So we look at an alignment, and this is an oversimplification of this idea. So we have a position where you see these residues, another position where you see these residues, and what you see is that in fact you have a local conservation. So So in the extreme case here, you switch from one negative to another negative, and you switch from one positive to another positive residue. And here you switch the charge event. But you do it in the entire subfamily simultaneously. And the suspicion is that this may relate to these two being in contact. You have an event that is correlated simply by the force of similar 3D constraints. You can use that to predict contacts between inside of proteins, between residues, and essentially predict protein 3D structures. Shown in a Cell paper, that was the first time that people really predicted structure from sequence. For proteins at that point when they published it, Thomas and Debbie and Chris Under, when they published that, these structures were not known. Meanwhile, some of these structures are out, and essentially what they did is really right. The problem in the method is they can do that only for some subset of all the proteins. They did this for membrane proteins here. They then essentially ran the method on any membrane proteins that they could lay their hands on, and they could predict for in the ballpark of 25 to 30 families, okay, of all the proteins from all the organisms. So it's amazing that you can do it for 25, but it's not really a big number, given that in human we expect something in the ballpark of 3,000 to 5,000 membrane proteins alone in human, right? But still, it's an amazing breakthrough, but a breakthrough that is limited simply because they need huge alignments in order to make this information, to separate the noise from the signal here. Now, the principal idea is use the same to predict protein-protein interactions. And there is a variety of methods that were proposed initially by Alfonso Valencia's group. Thomas and Debbie advanced it way over the initial way, but the idea essentially is old. Now, wait a minute. The mirror tree is sort of a related idea. I will skip over it. You essentially look at the similarity of two trees, and from the similarity of two trees, you try to infer that the proteins interact. Idea 5 somehow is more directly what you said before. You use motifs directly that are known to somehow relate to protein interaction. So you see a particular motif on a protein, and that motif relates to the interaction, then you essentially say that these proteins interact. Those are sort of the 5 simplest existing methods, and they all get somewhere. And they all are not they are not applicable to many cases. I briefly mentioned this idea of arrays. We can use array data. So co-expression relates to protein interaction prediction. If you use machine learning in between, that's a longer story. Ultimately what that says is directly you cannot use correlation microarray data, but you can train a machine learning device to actually make it useful to some extent. Idea number 7 is to use structure, 3D structure. So there the idea is if you essentially have a similar 3D interface in a different protein, and you essentially predict by homology modeling the interface and the match of the interfaces, and then you use 3D structures to predict. And that was introduced, here's the relatively recent Nature paper by the group of Barry Honig at Columbia. And they showed a few examples where they predicted structures or interactions that nobody had ever seen before. That's somehow what they estimated how well they do on a false positive rate versus true positive rate, versus in correlation to sort of other experimental and predictions, remarkably high. But whatever, they predicted many things that were clearly not known before. So there is a bunch of methods out there. Back to sequences only, I believe the best solution there is at the moment the profile kernel SVM that I discussed about. The overall accuracy is fairly low, and that brings up the next issue. Can we find a way to simply integrate all of these simple prediction methods and use the best of all data types, and one way to integrate is naive Bayes. In this naive Bayes, let me not get into that. And that's what most people do. And I believe, so originally, now I came into the issue of predicting protein DNA interactions. That is very similar to predicting interaction sites on a protein. Same idea of a sliding window. You can predict protein RNA, you guys doing it. The major issue is that if you only use the known DNA and RNA binding proteins, you overestimate the prediction, so you need negatives in your data set, and the negatives cannot be not seen in the data set, so the inversion for protein DNA binding like that, I take my data set and what I haven't seen is negative, doesn't work in that case. You also need to find really proteins that absolutely do not bind DNA, so a set of different proteins. Now, I glossed over one final issue here related to a problem with protein-protein interactions, and I'm not entirely sure how to formulate that. Cross-validation or negative selection or whatever you say. Here's what I did. Here's what we did. The positives, so again, goal was to predict that A and B bind or not, binary, not where they bind. Positives, we chose PDB, because PDB is really the most accurate. If I want to know whether two proteins bind, and if I know it on the mechanical detail on a per residue base, this is the most accurate way I can get. The negatives, we essentially chose from Swissprot or Uniprot, because we argued that, very much in the spirit of what we discussed before, not seeing it in the PDB does not mean it doesn't exist. But if nobody has ever seen it by any experimental method, then that is a good starting set to say they don't interact. So that's a negative. That means that, so this is Swissprot, because most of the proteins for which people do these two hybrid or TAP experiments simply are not in PDB. Okay. And that got us to what in my keynote talks I call method number 41. And method number 41 was amazingly accurate. And then Tobias realized something. What do you believe could have happened? So something went wrong. Can you suspect what happened?